So, I hope you understood the terms genotype, phenotype, alleles, homozygous, heterozygous, dominant and recessive. And I mentioned one scientist's name in the previous class, that is Gregor Mendel, who is considered as the founder of modern genetics, and he has done many experiments to find the pattern of inheritance. He has done most of his experiments in garden pea plants, and he took one character first, that is the height of the plant. And he found there are only two alleles that is controlling this gene for this character. They are, one is capital T, that is for tall plant, and then small t, that is for dwarfness or dwarf plant. So you can refer page number 351 for this one. So when he took one character and to find out the pattern of inheritance, we call it as a monohybrid inheritance. We are going to start with this one, monohybrid inheritance. So, which is the character that he has selected? He selected the character height and he found there are only two alleles that are controlling the character. Capital T is for tall plant and small t for dwarf. So you can refer page 351, which picture it is given. So he selected two plants. One is homozygous tall. So there is a way to write this one. First we have to write phenotype of the plants. So he selected one tall plant and then one dwarf plant. Okay. And the genotype. Genotype is, it is pure breeding. Pure breeding means there is no adulteration. That means both the alleles are same, or we can say homozygous. Homozygous tall and homozygous dwarf. There is no need to say homozygous dwarf actually, because dwarfness will be expressed only in homozygous condition. Okay. Now, this is diploid cells. Each diploid cell, this will be the genotype. And you know that during gamete formation, what happens? It will become, the cells becomes haploid. That means the alleles will get separated. Chromosome will get separated along with the alleles, so that the cells will have, each cell will have only one set of allele. And you know that here both are same only, so you can just write only one set. Maybe you will get mistaken that capital T should be big. No, it's like this. So these are the alleles that are present in the haploid cells. It's haploid, you know, this is diploid. Two sets are there, and when the gametes are formed by meiosis, each chromosome will get separated along with the alleles, and one allele will be going to each cell. Okay. So, when they are fertilized, that means they become a diploid cell. What will be the genotype of the first progeny? This is this one. Capital T, small t. So you know that whether it is haploid or diploid. Since there are two alleles present for the same character, so it will be diploid. Two sets are there. So all the F1, what is the genotype? The genotype is capital T, small t, and the phenotype is tall. Okay. Then what he did is, he selected this first progeny. That means all of them have the genotype capital T, small t. and then he allowed them to do self-fertilization. Okay. So, two plants, both of them are having the genotype capital T small t, and the next one is also having genotype capital T small t. So, how many different gametes can be produced from here by meiosis? There are two. Half of the gamete will have capital T and half of them will have small t. Here also, half of them will have capital T, half of them will have small t. Now we will do the crossing. Okay. So at the time of fertilization, any gamete can join with any other gamete. So it can be either this capital T or capital T small t or capital T small t or finally small t small t. So how many offsprings are there? 4 offspring. And what about the phenotype? How they are, whether they are tall or dwarf. This is tall, this is also tall, the third one is also tall, and the fourth one is dwarf. That means the phenotype is 3 of them are tall and 1 is dwarf. So we can say 3:1, or we can say 75% is to 25%. It is the phenotype. Means from outside you are seeing 3 of them are tall. But you know the genotypic ratio will be 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. Okay. So instead of doing this way, we have another way to do this crossing. Okay. Just draw this, it is known as Punnett square. Okay. So there are 4 boxes. Here we have to write the first gamete, second gamete, and the other parent, first gamete, second gamete, and you can cross it. Capital T, and capital T small t, capital T small t, small t, small t. So here also you can see same thing. The same thing I have done with the grid, and this is known as Punnett grid. Okay. You can refer page number 351. So you can see the same thing, like 3 tall and 1 dwarf, but genotypically it's homozygous, 2 heterozygous and 1 homozygous dwarf. So is it clear? So here we are saying it as monohybrid inheritance because we are selecting only one character. Okay, and there is a law of, for this one it's known as law of segregation. Law of segregation. Law of segregation states that at the time of gamete formation, each allele is separated and going to segregations, separated or segregated and going to different cells. See here the alleles. Here also you can see both the alleles. Yes, see 2 alleles are there at the time of gamete formation or meiosis. Each allele is separated and going to different gametes. So that is why it's known as law of segregation. With that, we are finishing monohybrid inheritance. Okay, hope you understood this. Then I will give you the test scores in the next class.